Great. Thank you everyone for participating today in the bipartisan Congressional House Cancer Caucus briefing. We're very happy to have you, and I'm going to kick over for opening remarks to Rachel Pollack.
Good morning, everyone. Thank you very much, Margarita, all for taking the time on this very busy September morning to join us for this important discussion. I am Rachel Pollack, Vice President of ACT for NIH. We are a very small advocacy organization dedicated to raising awareness around the vital life-saving research done by the National Institutes for Health, and advocating for robust and consistent appropriations for NIH. This morning, we are deeply honored to partner with the House Cancer Caucus, led by Representatives Kilmer, Higgins, Fitzpatrick, and Kelly, two of which are able to join us this morning, and the American Society for Gene and Cell Therapy to bring you this discussion of what's next in cancer therapy and research. I'm delighted to kick things off by introducing Congressman Brian Higgins, who has been a steadfast champion in the fight to end cancer as we know it. Representative Higgins?
Thank you, Margarita and Rachel. Thank you for joining us today for this important briefing on innovation in cancer care. Over the past several decades, we have made significant progress in the fight against cancer. As of 2018, the cancer death rate for men and women combined has fallen 31%. This decline represents roughly 3.2 million less deaths. Immunotherapy, CAR T-cell, these specialized treatments offer hope to many patients. CAR T-cell can result in complete elimination of the disease. This progress shows we may be close to a cure for cancer. There are now 6 approved CAR T-cell therapies in the United States. The science and technology behind them is the result of decades of research and sustained federal support and the hard work undertaken by institutions like Roswell Park Comprehensive Cancer Center in my district in Buffalo, New York. Federal dollars come from the National Cancer Institute and the National Institutes of Health that are critical to getting these therapies from discovery all the way through approval. And as co-chair of the House Cancer Caucus, I have long pushed for increased funding for cancer research. I invite all of my congressional colleagues to join me in the ongoing effort to ensure that we are investing in research that will ultimately save lives. We are fortunate to have an esteemed group of speakers today. Dr. Hans-Peter Kiem, Director of Stem Cell and Gene Therapy at Fred Hutchinson Cancer Center and the current President of the American Society of Gene and Cell Therapy. Dr. Rayne Rouce is an Associate Professor of Pediatrics and Hematology at Baylor University Medicine, and Dr. Renier Brentjens, Deputy Director and Chair of Comprehensive Cancer Center, Roswell Park Cancer Institute, and Rachel Pollack, the Vice President of ACT for NIH. I want to thank all of you and the panelists for lending their expertise to discussion today. I also want to thank ACT for NIH and the American Society of Gene and Cell Therapy for working with us for this event. Before we hear from our panelists, I'd like to turn things over to my colleague and fellow Cancer Caucus co-chair, Representative Brian Fitzpatrick.
Thank you, Brian. And good morning to everybody. It's an honor to be with you today to speak on a disease that we all know by virtue of our membership of this caucus impacts every single American and certainly impacted our family. And I wanted to thank our speakers for being here with us today to educate us specifically about immunotherapies. An immunotherapy known as Keytruda, which I know you're all familiar with, gave my family, several extra years to be with my brother who passed away from cancer. So it's an honor to be with you today to talk about this. As you all know, cancer is the second leading cause of death in the United States, and COVID-19 has only heightened that statistic. The pandemic, amongst all the other challenges that it posed, it also made access to treatment and early detection even more difficult. And as a result, cancer mortality rates are now rising, unfortunately. And it's critical that we act quickly to combat that aspect of this fight against cancer, the impact that COVID-19 is having on the cancer community, and also to ensure the patient access remains robust and the patient experiences are as positive as they possibly can be. Additionally, long-term planning is needed to be put in place in order to ensure a steady funding stream is given to cancer research. Current federal funding levels for cancer research do not match the rate at which people are suffering and dying from cancer. I've introduced several pieces of legislation, one of which is called the KO Cancer Act, to provide a once-in-a-generation massive infusion and investment in cancer research funding, which would increase cancer research funding and that allocation to NIH by 25% above and beyond current levels. Moreover, we cannot forget the impact that cancer has on children when having this discussion. That is perhaps the most painful thing to ever experience, watching a child suffer through the pain of cancer, and no parent should have to watch their child struggle to survive either, which is why another piece of legislation that I've been pushing, it's called the Fairness to Kids with Cancer Act, which enhances federal funding levels specifically for pediatric cancer research. And this legislation, amongst other things, takes steps to ensure pediatric cancer researchers have the funding necessary to save as many lives of our children as humanly possible. And as is the case for so many in America, as I mentioned, this fight is personal to me. It's personal to so many people I know. We must not stop until we eradicate this disease once and for all, and spare parents, spare children, spare caregivers who endure incredible mental anguish just caring for cancer patients, as well as families, the pains of cancer. So thank you. Look forward to having you. Brian, thanks for having me. And I yield back.
Is Mike Kelly on? Any other members want to offer something before we start the panel discussion? Okay, why don't we kick it off?
Do we start now, Margarita, or?
Yes, we're just getting the slides pulled up.
Okay. In the meantime, good morning. Yes, so my name is Hans-Peter Kiem. I'm the president of the American Society of Gene and Cell Therapy and also professor here at the Fred Hutchinson Cancer Center in Seattle. And I think we're just waiting for the slides to come up.
They're up on the screen now.
Oh, wonderful, yeah. So I just want to talk to you today a little bit about gene and cell therapy, to provide the basics. And certainly the members of our society, the American Society of Gene and Cell Therapy, have developed and provided the foundational technology for this next generation engineered immune cells and cancer therapies. So what is gene therapy? It's really when we deliver genetic material, DNA, RNA, to the cells, either inside the body or outside the body. Most of it occurs, of course, right now outside the body, where we insert, small pieces of genes like a chimeric antigen receptor, as I will show you, that will then make a CAR T cell or even correct defects in the genome. As you can see there, we can simply swap out different defective nucleotides. So it's a very exciting field, and we can use that technology as well to empower the T cells to make CAR T cells even more powerful. Next slide. So how do we make these chimeric antigen receptor T cells? This is just shown here. Typically, as I mentioned, we do this outside the body. We take the cells from a patient, the T cells, bring them to a laboratory, and then insert this chimeric antigen receptor into the T cells. And I'll talk a little bit about how we do this in the next slide. But then we grow these cells to millions of these CAR T cells, and then if you click forward, and then infuse them back into patients, where then these millions of immune cells really find specifically the cancer cell, and you can see this here, and detect the cancer cell very specifically and are enabled to then kill the cancer cell. So very powerful technology. So how do we insert now these CAR chimeric antigen constructs into these T cells. In most cases, and if you click forward, in most cases, we use a viral vector. And of course, everything in this viral vector, all the viral genes have been removed. So it's really like a little Trojan horse. We just put the chimeric antigen instructions into this shell, this Trojan horse, and then deliver this to the T cell to modify the T cell and empower the T cell to make this chimeric antigen receptor and then detect the cancer cells. Or there's other ways to do this as well. You may have heard about nanoparticles, in particular, of course, the lipid nanoparticle technology that has been developed and has been used, obviously, for COVID. So very similar to what is being done for the COVID vaccines. We can use these nanoparticles now also to help them deliver these CAR constructs to patients and, again, empower the immune system to fight cancer that way. Or we can be very, very precise, and this is the most exciting field over the past probably 5 years, and over the next 10 years, it will be the most exciting field, I think, the gene editing technology that enables us now to very precisely modify, as you can see here, really single bases, very precise edits in the T cells, in the DNA of the T cells, to really make these CAR T cells now much more powerful and much more durable to hopefully, improve their ability to fight and kill cancer. Okay. Now, I think this is my almost last slide. So in terms of the pipeline, you can see most gene therapies currently in the clinic target cancer. This is that upper bar there, 376 number of therapies that are in investigational studies in the clinic currently for cancer. And as you've heard on the next slide, there are now already 6 CAR T cell studies approved. Currently, those are the different CAR products, and over 5,000 patients have been treated now with Kymriah, and more than 4,600 patients have been treated with Yescarta, and the results have been very, very favorable in many cases with durable responses. And I think you'll hear more about this from my colleagues. Thank you.
Good morning, everyone. I'm so excited to be here to talk to you about the new face of cancer therapy. And I'm going to specifically talk about delivery of CAR T cell therapies and the patient experience. As we mentioned, I'm Dr. Rayne Rouce. I am a pediatric oncologist. I was very excited to hear some of the committee's efforts to enhance the therapies we have for children. Next slide, please. So this is called a survival curve. This is a graph that may look complicated at first glance, but it's actually pretty simple. So on the left, we have percent survival. And obviously, when we're talking about survival from any disease, we want it to be closer to 100%. And on the bottom, we actually have years since achieving a complete remission. So when I started taking care of children with cancer, particularly leukemia, which is the most common childhood cancer, if patients did not respond to initial therapy, their survival curves were dismal, as demonstrated here. Only about 8 to 10 years ago, we started to change the way we actually treated childhood cancer. Next, please. And CAR T cells came on the scene and literally have been groundbreaking. So CAR T cells for the most common childhood cancer, acute leukemia, kids and families that literally a year prior to that, I would have been saying that we had exhausted all treatment options. Now I can say there is an 80% chance that you will be cancer-free after a single infusion of CAR T cells. And importantly, next, I can actually say that there's a 60% chance that you will remain cancer-free 1 year later and beyond. Next slide, please. So this is revolutionary. It's literally life-saving for so many patients, but the road to access to these CAR T cells on the part of the patients, on the part of the providers, truly is an uphill battle, and it's littered with roadblocks along the way that they really have to overcome. Next slide. So Dr. Kiem did a great job of explaining what we actually do in the laboratory to make these cells, but I want to take a step back and focus on the patient experience during this time point. Next. So even before we're able to remove blood from the patient to actually manufacture these T cells, there is a lot of preparation that's involved. The patients and their referring oncologists have to identify a CAR T treatment site. And in many cases, this can be somewhere outside of the state. It could be hundreds of miles away. Then they have to obtain insurance approval. And despite the fact that some of these CAR T cell therapies have been FDA approved for 5 years now, this process can take months. And these are months that these heavily pretreated refractory cancer patients do not have. Then we have to coordinate T cell collection. And once we have removed the blood from the patient to generate the CAR T cells, we have to ship the CAR T cells to the manufacturer. So this is really exciting science, and it may seem like a short period of time that the making of these CAR T cells can take only about 8 weeks, but the issue is during that 8-week period, next please, there is quite a balancing act of trying to control the patient's cancer while limiting the toxicities from additional therapies that they truly need to keep their cancer under control while they're waiting that 8-week period. And so we often run into a situation where we know these CAR T cells are coming and we just have to keep that patient with us and healthy enough. And it requires us to give additional therapies that are often futile when it comes to actually curing the cancer, but certainly lead to additional toxicities. And the last step, when we're excited and we get word that the CAR T cells are actually ready to give to the patient, we have to do tons of planning. So we have to arrange for patients to travel to the CAR T site, which again is often out of state. These patients typically have to remain within the vicinity of the CAR T cell center for at least 4 weeks after the infusion of CAR T cells, often in the hospital. So you can imagine whether this is an adult who has to have a caregiver with them or whether this is a child who has to have a parent leave, the siblings, it is completely disruptive to the families. Next slide. And at the beginning of this, at the center, if you'll go back one time, at the center of all of this, we have the cancer patient. And that's what I want to really impress on everyone, that this is really complex therapy that we've been able to do, but it can be extremely stressful for the patient. So we're really fortunate in 2022. We have lots of CAR T cells that actually work for blood cancers. We have been able to cure people who previously had a less than 10% chance of living. Next slide, please. As Dr. Kiem mentioned, we have 6 FDA-approved CAR T cell therapies for leukemias, lymphomas, and myelomas. And so many of us are working to expand CAR T cells beyond blood cancers. And while this has been a greater challenge, we are scientists and we're not afraid of a challenge. And Dr. Brentjens is going to talk about some of the new and exciting therapies coming down the pipeline. And importantly, we've established CAR T centers of excellence. We've partnered with patient advocacy and scientific societies such as ASGCT and have risen to the challenge to ensure that as we deliver these complex therapies to patients, we're keeping safety in mind and we're making it easier for patients. We're working on shared care models that allow some of this treatment to occur closer to home so it's not quite as disruptive to the family's lifestyle and community. You shouldn't have to trade your comfort for a chance at a cure. But sadly, we are still unable to get these therapies to many underrepresented and under-resourced patients who may live within your districts. And this is not just because of cost and logistical constraints, it's because it's a very complicated process and time is of the essence. So we really need to get to a point where we can break down some of these barriers. This is my last slide, and I think a perfect setup for Dr. Brentjens to tell us about what's new on the horizon. Thank you so much.
Okay, so I actually don't have slides. So I think that I was asked to talk about what the future of this type of therapy is. And again, I go back 24 years. I was at Sloan Kettering working with Michelle Sadelain. We did the first published studies with CAR T cells in adult patients with acute lymphoblastic leukemia. And I think what you can appreciate from the 2 prior presentations is that this is a technology that's still in its infancy. We are going to have to expect the big pharmaceutical companies to improve the delivery, not just time-wise, but also location-wise of these life-saving therapies. But the other thing to appreciate is that there are 6 FDA-approved drugs, and they are all for B-cell cancers. So that's leukemias, lymphomas, and multiple myeloma. And that is for a reason, because immunologically, these tend to be less complex cancers. And the question that we as investigators are faced with currently is this a proof of principle, or is this simply a better way to treat B-cell malignancies? And I think everyone, all the panelists here would agree that it's a proof of principle. The concept of moving this technology to solid tumor malignancies, which are far more common, cause far greater numbers of deaths, is very complex. And it's complex because these solid tumors tend to not all express targets that we can go after with these CAR T cells. And in addition, these tumors scaffold themselves with other cells that suppress the immune system. And it's difficult at times for these T cells to actually penetrate into these tumors to be able to eradicate them. And what that has led to is a burst of innovation coming predominantly from academia, where we try to grapple with how we can fit that model that works so well with B-cell and blood cancers into far more common solid tumor cancers. We are one group that have pioneered what we call armored CAR T cells, where we do additional genetic modifications, not just so that the immune cell can target the cancer cell, but when that immune cell gets to that tumor, it can modulate and change that microenvironment that is so immune suppressive to being one that is very pro-inflammatory to allow for this technology to work. And in addition, these cells now have the capacity to try to recruit a patient's own immune cells to better target the cancer cells. And so what one would predict is that this CAR T cell technology will successfully move into solid tumor malignancies. Now, how that's going to look is yet unknown. It's a little bit right now like having a flip phone and trying to predict a smartphone. And so it'll be interesting to see 5 years from now what the design is on this basic CAR T cell platform. And so what I often like to tell when I give talks is I have a slide with a Model T Ford and a Ford Mustang, where the Model T Ford is where we are right now, and the Mustang is where we want to go. And so that requires a significant amount of NIH funding. I would point out that when we started out with CAR T cell therapies back 24 years ago, it was NIH funding that allowed us to get to where we are today. It was cooperation with the FDA that allowed us to get to where we are today. This was all very high risk when we started it. And obviously, it's very satisfying for us to see that it now is causing so much excitement. What this means, however, is that the issues that were brought up are only going to get exacerbated as we get more common tumors and we have CAR T cell therapies that work in these more common tumors. And so over the next 5 to 10 years, things are going to get very exciting, but we will have to use the platform that we have now for B-cell cancers to refine it in such a way that when we get these technologies to work in more common tumors, that we also have the infrastructure in place that we can provide these cells to patients in a timely fashion, as well as provide these therapies to patients that are otherwise in regions where access may not be as readily possible. The final point I will bring out is that these are very expensive trials to run in an academic center. And so what that has led to is a significant marriage between industry and academia. And that is going to be significant moving forward. To run these initial phase 1/2 trials, these early stage trials, cost a significant amount of money, and most investigators There are no NIH-based mechanisms to pay for such trials. And so most investigators, including ourselves, are dependent on developing relationships, whether it's with small startup companies or whether it's big companies like BMS, Gilead, et cetera, to help fund these trials, to move these technologies forward, successfully forward to the clinical setting. And I think I will stop there and simply state that this is in its infancy. Despite 6 FDA-approved products, it's going to be both very exciting and very harrowing to see where this goes in the next 5 to 10 years. And we do hope that ultimately this replaces chemotherapies that are so toxic to our patients.
Thank you, Dr. Brentjens. Rachel, Pollock, I think you're up next.
Good morning again, everyone. Thank you so much to all of the speakers. You've done such an amazing job sharing your work that brings hope to so many patients and their families that are struggling with cancer. However, as we all know, life-saving research requires federal funding. Each year, about 80% of the appropriations made to the National Institutes of Health goes back into states and communities in the form of research grants to universities, research institutions, and small businesses. These grants are extremely important in the search for cures and treatments. And it's important that we advocate for robust and consistent funding in order to get to more grants for the National Institutes of Health and especially the National Cancer Institute. As you all also know, this week, Congress will pass a continuing resolution to fund the government through the election. And after the election, Congress will return to finish the work of the FY 2023 appropriations process. I just wanted to give a little bit of information about where things stand in that process in the cancer space. So right now, the House Labor and Health and Human Services Appropriations Bill for FY23 includes a $2.5 billion increase for NIH and a $466 million increase for NCI, the National Cancer Institute. The Senate Labor H bill includes a $2 billion increase for NIH and a $290 million increase for NCI. The National Cancer Institute professional judgment budget for FY23 requests an increase of $1 billion. That's what they say they need in order to continue this life-saving research that all of our panelists have talked about today. So we're encouraged by these increases and by the numbers provided in the House and the Senate. We look forward to seeing how these appropriations translate into more and more life-saving treatment and more hope for these patients and their families. So thank you, Margarita, and I look forward to listening to all the questions and answers we might have today.
Great. Thank you to all of our speakers. I just put a note in the chat. If you have any questions for the speakers, please feel welcome to either put it in the chat or you're welcome to unmute yourselves and to ask. We have really an all-star panel of experts here, and we'll share the recording of this briefing with you. We will actually stop recording now.
And again, thanks to all of our guests. I think many of you likely know last year the House Cancer Caucus recognized the 50th anniversary of the National Cancer Act, which really changed the way we understand and study and treat cancer. It established a network of cancer centers and clinical trials. It established the National Cancer Institute as we know it today, and drove really historic investments into cancer research, leading to revolutionary new treatments like CAR T therapies that we discussed today. So these individualized targeted therapies really reflect the successes of sustained federal investment in cancer research, and I think bring new hope for Americans with cancer. Our discussion today, though, also points to the continued need for additional federal investment in cancer research and far more equitable access to cancer care. Despite advances that we've seen in care, 1.8 million Americans will be diagnosed with cancer this year. 600,000 Americans will die from cancer this year. And those aren't just numbers. Those are people we care about. Cancer affects every family in America, including mine. I lost 3 of my 4 grandparents to cancer, and I feel lucky that other members of my family are still alive as a result of advancement in detection and treatment. Sadly, we lost a member of my district office, a young woman in her 20s, to cancer just a few years ago. And I think about her nearly every day, and I think about her family. And unfortunately, we also know that the pandemic has created some new and continuing challenges in treating and studying cancers. Many Americans didn't get routine screenings during the pandemic, and providers are reporting that patients are presenting with some more advanced or complex cancers. And cancer research has hit some roadblocks during the pandemic. Research labs were disrupted, projects were delayed or canceled, clinical trials faced some new barriers. And I think it's why not only am I committed, the Cancer Caucus is committed to supporting funding for cancer research at NCI, at cancer centers around the country and elsewhere. It's why we're committed to working with the White House's reignited cancer moonshot to achieve President Biden's goal of reducing the cancer death rate by 50% over the next 25 years. And a lot of that just means supporting cutting edge research at NCI and elsewhere to spur that innovation, to improve our knowledge and understanding of human health, and in turn to provide hope for cures and hope for treatments and hope for families. So again, I want to just thank our guests and thank all of the members and staff members who were able to be on and watch. And we got a lot of work ahead of us. And so let's get to it. Thanks, everybody.
Thank you.
Thank you.
Thanks.